Hola amigos and welcome to What's Your Question? Today we're going to be answering the question, what is gene therapy and why do we need to know all about it? Let's go! Before we start off, I'd like to have a couple of questions. I've placed them here for you to have a look at. The questions are, what was the Human Genome Project? And to name 2 examples of genetic disorders. I'd like you to pause the video here, have a think about it, write it down if you can, maybe put it down in the comments, but you need to have a think about the answer to those 2 questions for me, please. Pause the video here, have a think. Fantastic. So, first of all then, the Human Genome Project you should have got as being a 13-year study that was done between 1990 and 2003, where they were looking to find out exactly what each part of the human genome is, what it does. They looked at all the genes and found out the jobs of 92% of those genes, tried to find out exactly what it is they were responsible for as you developed as a human being. 2 examples of genetic diseases you could have chosen here were Huntington's disease, where it causes your mental capacity to degrade over time, and also you could have mentioned cystic fibrosis, which does have an effect on the lungs. I've placed the objectives for today's lesson here for you to have a look at. The objectives for you to think about today are, first of all, how do we use gene therapy? What are the different ways that we use it? Then we're going to move on to think about exactly how gene therapy works. What do they do to insert a gene into a living organism? And then we'll talk about the risks and benefits of gene therapy, whether it's worth us doing it or whether we should avoid doing it. You can consider and decide, make your own opinion based on the risks and benefits that we talk about today. So to start off with then, how do we use gene therapy? What are the different ways this is useful to us when it comes to treating people? The first of these fits into embryology. Embryology is us using embryos, so unborn children, in the initial stage of development and very just after fertilization. A fertilized egg cell will reproduce and become a small ball of cells made up of only stem cells, and this is an embryo. When IVF is being carried out, they will make a number of these embryos and store them for use during IVF. If successful, when it comes to trying IVF, in vitro fertilization, we're trying to make it so that somebody has a baby, then they will not use all of those embryos. So these embryos will never become a fully grown human being. So these are often then used in screening trials to check them and look at their DNA and find out different information about them. And we can study how different cells are affected by different things, how different diseases affect different cells, and so on. So it's really useful when it comes to that, as we can then research how we can use gene therapy to better treat people using those embryos. We can also use this in what's called a saviour sibling. This involves when somebody is going to become very, very ill from a genetic disease. They will try and find a donor for that person. This generally works best in children. And if they cannot find a person in time, they can use embryos or embryonic screening to check the DNA of an embryo to make sure that they are compatible with the person that needs for example, a new organ or specific cells, and they will then be able to deliberately cause that embryo to grow into a baby by implanting it into the mother, and then use the cells from that baby when it is fully grown. So they can use this genetic information to best predict what organs are going to be formed and whether they'll be compatible with a person, and then make the ones that they need to make. We also have designer babies during IVF. We know that we are deliberately causing one sperm to fertilize an egg cell. We have pre-implantation genetic diagnosis. So in this case, before a sperm or egg cell is implanted into the egg, or before the embryo is implanted into the mother, they can screen the embryo to make sure that it doesn't have any of those genetic diseases that the parents may have, that siblings may have. So if one sibling has cystic fibrosis, for example, the next one can be checked to make sure that it won't have that when it's born, and they can pick the specific embryo that's going to be the most healthy using this technique. It is very, very expensive. but that's one thing we can do. We also have pre-implantation genetic screening. So in this one, they're just checking to see whether it's male or female. As far as I know, they don't do this one particularly often. And this one can be that before it's implanted, you can pick whether it's a boy or a girl. We also have germline therapy. In germline therapy, they can change the sex cells in organisms. Again, ethically, not one that we do very much, but I think in some countries it's something they do. And finally, we have somatic cell therapy, and this is where we're changing the cells in a fully grown human being, and that means it will only affect the human being themselves and not their sex cells and not any of their offspring. So it'd be about changing the genetics of a person that was already born and not affecting the children that will be given off. So those are just a few different ways that we can use gene therapy when it comes to treating diseases. Objective 2 is all about how we go through the gene therapy process. And there's only really 3 steps to this process. All they're going to do is, first of all, find somebody with the gene that they require, with a normal gene that they need. So, for example, if it's a person who's going to get Huntington's disease, they find a person that's not going to get it and extract the gene that is responsible for this. They then make multiple copies of this. They would extract that gene in the same way they would do when they're doing genetic engineering, using a restriction enzyme to cut that out. They then, once they have got it, and they've made lots of different examples, they would implant that using the ligase enzyme into a virus, because viruses have a really important skill in that when they infect a specific cell, they can change the DNA of that cell. So once it's implanted in the virus, all you've got to do is then use the virus and inject it into the person, and that virus should then go around and affect the cells of that organism they're injected into. So that should then be able to be used to treat that illness. So the steps again, they're going to isolate the required gene, the normal gene. They're going to replicate that gene to make multiple copies of it, and then they're going to add it into a virus to be used as a vector, which means it can be transferred from one organism to another, and then inject that into the person so the virus can implant that DNA into the person they want to implant it into. Finally then, we have the risks and benefits of gene therapy. So the benefits of gene therapy are that we can use this process to change the DNA of these things to make them more useful. Good examples of this are we've changed the DNA of some bacteria so they can produce insulin that we need. Millions and millions of people around the world are born with type 1 diabetes and require injecting insulin into themselves, and this is the best and biggest scale way of creating insulin on a level that we can then use to treat those people regularly without any issue. It can prevent the onset of specific diseases. We've talked about how we can use this to edit the DNA and stop those things from happening. They can change the DNA, make it so they're not likely for those diseases that would normally start off to kick off in the first place. They can cancel them out completely. They can remove diseases from people that are born with those diseases. So if you're born with something that makes you very ill, we can use gene therapy to extract those genes and replace them with normal ones. And finally, we can fix unwanted mutations inside those people as well, and make sure that they are able to get better from those mutations, or they don't affect them and cause negative effects on their lives as a whole. The risks of this are quite big because it's not something we researched in a huge way. We've not tried it lots and lots of times at this point. We are still completely uncertain about the overall way it can affect a person or an organism. We haven't done it a huge amount of times, so we're not 100% certain what effects it can have. The things we do know so far are it can cause us to have an unwanted immune system reaction. It can cause us to react in a way that we wouldn't be able to predict, because these are foreign cells infecting our body. Our immune system can then try and fight these off and can make us very ill if it does try and do that. So it's really important that before we do these things, we make sure that the people that we inject them into aren't going to have serious responses in that way. We also need to make sure that the viruses involved are completely deactivated, because if for any reason that virus is not deactivated when we inject it into that person, obviously the virus is still going to have the normal effect that it was and could again lead to an infection in some way from that virus, which could be massively negative in terms of if they're already fragile, it can make them very ill very quick. So making sure that the virus is not something that can cause them to become ill would be essential to this, so there's a risk there. We've also found that it's possible for these to cause a tumour in some cases, so we need to make sure that isn't going to happen in these cases also. We also need to consider when we're doing this, those ethical considerations we've talked about before. Is it right for us to be editing the DNA of organisms? We know that a lot of religious groups do not agree with this and think that we shouldn't be able to do it. They think that we're playing God, and they don't agree with that, and they are vehemently against the use of processes like this one. I know that people like Jehovah's Witnesses will not even allow blood to be injected into their bodies if they're getting ill. So in terms of changing their actual DNA, it's not going to be something that's going to sit well with those people. Because of the use of stem cells as well, some people disagree with the use of them, and they think they're killing something that could become a living thing, and they disagree with that on a huge level. So ethically, there's lots of considerations to take into account here as well. But that's just a quick tour around how gene therapy works. If you have any questions, throw them in the comments, and I'll make another video answering any questions that you have. Thanks very much for watching. Make sure you stay safe, stay alert, stay curious, and subscribe.